Good morning, students. Let us study a few spots today. The first spot that we'll study is GPCR or G protein coupled receptors. They are also called as 7 transmembrane receptors. They are found in many organisms such as mammals, plants, microbes, and invertebrates. These receptors transmit signals from certain substances which binds on the extracellular surface to an intracellular molecule called as a G protein. G means guanine nucleotide binding protein. This GPCR has 3 basic regions. The first is an extracellular domain, which is the N-terminus, intracellular domain, which is C-terminus, and the middle segment, which contains 7 transmembrane domains, which means that this segment traverses the membrane 7 times in a serpentine pattern. The last domain is connected to the C-terminus. Now, when a GPCR binds to a ligand which possesses the affinity for the receptor, the ligand triggers a conformational change in the 7-TM region. This conformational change activates the C-terminus, which then recruits a substance which activates G protein associated with the GPR. The activation initiates a series of intracellular reactions that end in generation of some effect for the cell. For example, this epinephrine stimulated CAMP synthesis shows that epinephrine binds to this 7-TM receptor and which activates this G protein. This G protein further activates adenyl cyclase on the intracellular surface. This adenyl cyclase then produces CAMP molecule, and this CAMP then triggers many reactions inside the cell. The second spot is bacterial 2-component system. In these bacterial 2-component systems respond to changing environments by altering the gene expression. These are also mediators of signal transduction, which enable bacteria to detect physical and chemical changes which are transmitted to the bacterial nucleoid, and modulation in gene expression occurs. Now, mostly TCS or 2-component systems have 2 proteins, a sensor histidine kinase and a response regulator. The histidine kinase is a membrane-bound sensor which senses specific signals or stimuli and leads to activation of kinase activity, and a conserved histidine residue is autophosphorylated. This phosphoryl group is transferred to a DNA binding response regulator which activates its activities. Now, these response regulators mostly are transcription regulators, and they turn genes on and off by driving dimerization and having high affinity to DNA. This happens because of the signals received from the sensor histidine kinases. They have many applications such as motility, nutrient uptake, cell division, pathogenesis, virulence, and since they are found in bacteria, they are considered as potential targets for developing new antibiotics. One of the most common 2-component system is the OMP-C system in bacteria. The next spot is structure of tRNA. Here you can see the 2-dimensional cloverleaf model and the 3-dimensional inverted L structure. If you can see in this figure, the structure of tRNA or transfer RNA has many parts. The first of them is the acceptor arm, where the incoming amino acid binds to this CCA sequence. Here you can see alanine is bound to this CCA sequence. So, since this arm accepts the incoming amino acid, this is called as acceptor arm. The next is T5C loop. It has unusual amino acid pseudouridine. The next is a variable loop which may or may not be present in a few tRNAs. The next is the anticodon loop, which has the anticodon which binds to the mRNA during protein synthesis. This anticodon loop is also responsible for the wobble positions, that what we study is the third the third base can be changed. Now the next is the D loop, or more specifically DHU loop, in which there is an unusual amino acid known as dihydroxyuridine. Now this tRNA is very important during protein synthesis, as it acts like a messenger between the amino acids in the cellular pool and the protein synthesis taking place on the ribosomes. Thank you.